Hey guys, I decided to make a video for muscle contraction to help you guys out for your last bio exam. Yeah, at first it was a bit far, so it might be kind of hard to see, but I kind of go through it step by step, so hopefully that makes it a little bit better. Yeah, I hope it helps. If you feel like being kind, I guess share it with your other bio 108 friends, and hopefully it helps all you guys out on your last test. It's been a lot of fun tutoring you guys this semester. I wish you guys the best luck with the rest of college. Okay, so first we're gonna do the sarcomere. So the sarcomere, you start off with your two boundaries. So this is gonna be your Z-line on the sides, and they're basically like the boundaries of your sarcomere, which is a subunit of your myofibril, which is in your muscle cells. So your Z-lines on the sides, and then what the Z-lines do is that they anchor your actin until your actin doesn't fly away. See if you guys can see that. Okay. Z-line. And then you want your M-band right in the middle. M-band, middle, and another way you guys can remember is that your M-band helps your myosin stay on the sarcomere. So this is going to be your myosin. Okay. So the overlap. This is your myosin. Okay. So you have your Z-line, M-band, myosin, actin. Okay. And then, now, what I left out was the protein that holds onto your myosin so it doesn't just fall down droopy this way, or that way, or that way, or that way. And this protein is called titin. Right here, and here. It is not lime green, by the way. Okay. So, the first band you guys need to know is the I band. And the I band, hope you guys remember from tutoring, is actin only. Actin only. And this band here, your I band, it's going to be one of your two no overlapping zones. And when I say no overlapping, it's because there's only actin and no myosin. And then your next one will be the A band, or A region, right here. A band. And if you remember from tutoring, your A band is just basically the location of your myosin. So whether or not contraction occurs or not, or if it doesn't occur, your A band should stay the same no matter what throughout. So your A band be constant. Okay. Let's see. What am I missing? You have your I band, A band. Oh, and the last thing is gonna be the H zone right here. H zone right there. So where your H zone is going to be your second no overlapping zone. No overlapping zone. And then the reason why it's no overlapping is because at all times your H zone is going to be composed of basically only myosin. So when contraction occurs and your myosin pulls the two actins on both sides closer in, your H zone is going to basically follow the ends of the actin in, and it's going to shrink during contraction. And during relaxation, they're going to spread out to the sides and get bigger. And then, so as a result, during contraction, when you have more overlap going on, your eye bands also shrink, because during contraction, your myosin pulls your actin in, so the Z line comes in as well, but the myosin doesn't move, so it's like this. So as a result, your eye band here, which is actin only, will decrease, because your actin is going to start overlapping more and more with your myosin. And that's about it for your sarcomere. So this is going to be a sarcomere. Okay. The next, muscle contraction. So this is going to be your brain. Okay. And your brain is going to basically send a efferent message down to a motor neuron. So that's your neuron. Your neuron actually is not as big as your brain, but here's the axon. The dendrite of your motor neuron is going to pick up the signal and turn it into a action potential. That was not me burping, by the way. The action potential travels along the axon until it reaches the axon terminal down here. So let's say this is your leg muscle. This is your kneecap. It's ugly. It's your feet. The neuron, it's actually much smaller. This isn't the right scale, don't follow it. But here you have the axon terminals. So now I'm going to draw this a little bit bigger so you guys can see that better. It's like the figure in your book. So this is going to be one single axon terminal right here, this little 1 millimeter thing. So it's going to be like this. This is the axon terminal. Let's see. Do it like this. So then this is your muscle cell. It's going to be the muscle cell, also known as your postsynaptic cell. And this is your motor neuron from before. He is also known as the presynaptic cell. Okay, so your action potential as it travels through the motor neuron, if you remember, as it travels through, it causes depolarization, and depolarization, most of the time, it's going to be associated with the opening of voltage-gated sodium channels. So yeah, at your, this part would be called the axon terminal. Your action potential is traveling down to the axon terminal. That's your action potential. When it reaches this point here, it's going to cause a membrane change in terms of voltage. So your voltage-gated sodium channels, because they're voltage-gated, so a change in voltage is going to open them up. Your sodium ions from outside goes inside your cell, and whenever sodium goes in, it causes depolarization. As a result of depolarization, because we're at an axon terminal, you also have voltage-sensitive calcium channels right here and here. So now calcium ions will go into your presynaptic cell, which is also your motor neuron. When they go in, your acetylcholine vesicles here, or whatever type of neurotransmitter we're dealing with. In this case, let's just say it's acetylcholine. This vesicle fuses with the cell membrane here, and then it gets exocytosed. Okay? And then as a result, it gets exocytosed into the synaptic cleft. This region, I'll just label it right here. Synaptic cleft, which is just like the space between your presynaptic cell, which in our case is a neuron, and our postsynaptic cell, which is a muscle cell. So then your neurotransmitters get released, and then in this case, our acetylcholine, it's going to bind to its receptor protein right here. If you remember from the figure in your book, 2 molecules of acetylcholine bind to each side of the receptor, and it turns out that your acetylcholine receptor is also a sodium channel. So when the acetylcholine binds to the receptor, sodium from outside of the muscle cell can now go in. As a result of sodium going in, you have a slight depolarization. And then once you have enough depolarization to cause the membrane potential to reach threshold, then all the neighboring voltage-gated ones, these voltage-gated sodium channels will now all open up. Over here, over here, and then you have, because they're voltage sensitive, once you reach that point, they all come in, and then you have instead of a slight depolarization, you have a strong depolarization. So with all the sodium channels coming, all the sodium channels opening up, and all the sodium ions coming in, it causes a strong depolarization, and then next you get a action potential. So let's see. I'm going to redraw this part over here. Now we're dealing with your muscle cell. So all the sodium comes in, and then you have an action potential that travels throughout the muscle cell. Okay, so action potential. This is a muscle cell. Let's see. In order to get the action potential all throughout the muscle cell, so your muscle will contract, you have structures called T-tubules. So they go like this, and then they literally bring the action potential down to the center of the cell. And then they go on here. So this network here, they call it T-tubules. Write it again here. T-tubules. And these T-tubules, they run along a structure here called your sarcoplasmic reticulum, which is basically just the endoplasmic reticulum of